In a sealed lab at Harvard, a geneticist opened a file that had been 3 years in the making. Inside it was the genetic code of 33 people who had been dead for 600 years, pulled from teeth buried under a parking lot in Germany. What that file revealed would force scientists to rewrite what they thought. They knew about one of the oldest continuous peoples on Earth. Because when researchers lined up that ancient DNA against the genomes of Jewish communities living today in New York, Tel Aviv, Casablanca, and Sana'a, they found something that should not have been possible after 2,000 years of exile, expulsion, and dispersion across 6 continents. A signal, a thread, a genetic fingerprint that refused to disappear. Every other empire from the ancient world has watched its bloodline dissolve into the populations that came after it. This one didn't. And once you understand why, you will never think about identity, ancestry, or the limits of history the same way again. For most of human history, the question of where a people actually came from was a matter of story, not evidence. Every nation, every religion, every tribe carries an origin narrative, and for thousands of years, there was no way to check those narratives against anything harder than other stories. That changed permanently once scientists learned to read DNA, not just as a blueprint for a living body, but as an archive of everything that body's ancestors had lived through. Every migration, every intermarriage, every population collapse, and every population boom leaves a signature buried in the genome. And by the 1990s, geneticists had the tools to start pulling that signature out of modern people, and eventually out of the bones of the dead. The Jewish people presented one of the strangest test cases population geneticists had ever encountered. Following the Roman destruction of the Second Temple in 70 CE, Jewish communities scattered outward across the ancient world and kept scattering for the next 2 millennia. Ashkenazi communities took root in the Rhineland and later spread through Central and Eastern Europe. Sephardic communities settled across Iberia before the 1492 expulsion pushed them into North Africa, the Ottoman Empire, and the wider Mediterranean. Mizrahi communities remained rooted in Iraq, Iran, and the broader Middle East. Yemenite Jewish communities developed in near total isolation on the Arabian Peninsula. Ethiopian Jewish communities, the Beta Israel, developed in the highlands of East Africa, cut off from the other branches of world Jewry for centuries. On paper, this should have been enough time and enough separation to erase any shared genetic thread entirely. Populations that scatter this widely for this long almost always dissolve into whichever larger population surrounds them. That is simply what genetic drift and intermarriage do to a dispersed group over dozens of generations. The question researchers wanted to answer was whether Jewish communities were the exception to that rule, and if they were, why? The first real crack in the mystery came from an unusually narrow and unusually testable piece of oral tradition. Within Jewish religious life, a hereditary priestly class known as the Kohanim has, according to tradition, passed its status from father to son for more than 3,000 years, tracing back to a single ancestor. That is an extraordinary claim to make about any lineage, let alone one that survived exile, persecution, and the total destruction of the institutions that gave the priesthood its original purpose. In 1997, geneticist Karl Skorecki and a team of collaborators decided to actually test it, publishing their results in the journal Nature under the title Y Chromosomes of Jewish Priests. Because the Y chromosome passes from father to son with very little change over generations, it functions almost like a genetic surname. And if the priestly tradition were biologically real, Kohanim should share a distinctive paternal signature, regardless of where in the world their families had lived for the last 1,000 years. The first study found a real and statistically significant difference between the Y chromosome markers of men who identified as Kohanim and the rest of the Jewish male population, and crucially, that difference showed up consistently in both Ashkenazi and Sephardic Kohanim, 2 communities that had been separated from each other for roughly 1,000 years by the time of the study. A year later, a follow-up study led by Michael Hammer and Skorecki's collaborators expanded the marker set and identified what came to be called the Cohen modal haplotype, a specific cluster of Y chromosome markers heavily overrepresented among men with a tradition of priestly descent. Later refinements using larger marker panels complicated the simple version of the story considerably. Subsequent research led by Hammer in 2009 found that the priesthood was not the product of a single unbroken paternal line, but of several distinct lineages, meaning multiple founding fathers rather than one, and that not all men with a Cohen tradition carry the original modal haplotype at all. The science did not confirm a fairy tale. It confirmed something more interesting than a fairy tale, a real, ancient, and statistically detectable paternal signal sitting underneath a religious tradition that oral history alone had preserved for around 3,000 years, exactly the kind of signal that should not survive that long in a population without something unusual holding it in place. The Y chromosome only tells half the story because it only follows the paternal line. To get the full picture, geneticists needed autosomal DNA, the DNA inherited from both parents, and ideally they needed it directly from ancient remains rather than reconstructed backward from living populations. That opportunity appeared in 2013 when construction crews in Erfurt, Germany, began converting an old granary into a parking garage and uncovered a medieval Jewish cemetery underneath it. Jewish law places strict limits on disturbing the dead, and it took years of careful consultation with rabbinical authorities before researchers received permission to extract DNA from teeth that had already become detached from the remains during the original excavation, a method chosen specifically to avoid further disturbing the burials themselves. The team that eventually analyzed the material, led by Shai Carmi of the Hebrew University of Jerusalem and David Reich of Harvard Medical School, sequenced ancient DNA from 33 individuals who lived in Erfurt's Jewish community sometime between the 11th and 15th centuries. The results were published in the journal Cell and immediately became the largest ancient DNA study ever conducted on a Jewish population. What they found surprised even the researchers who ran it. The medieval Erfurt community turned out to be more genetically diverse than modern Ashkenazi Jewish populations are today, splitting into at least two distinguishable subgroups, one with a stronger Middle Eastern genetic signal and one with a stronger Eastern and Central European signal. At least 8 of the individuals buried in that cemetery already carried disease-linked genetic variants that are today considered hallmark markers of the Ashkenazi founder event. Mutations associated with conditions like Tay-Sachs disease and certain BRCA1 variants, meaning the population bottleneck responsible for those elevated disease frequencies had already happened by the 1400s, earlier than many researchers had previously assumed. As Carmi put it, modern Ashkenazi Jews from opposite sides of the planet are today almost genetically indistinguishable from one another, but 600 years ago, that same population was still visibly more varied. Somewhere between medieval Erfurt and the present day, something compressed that diversity down into the remarkably uniform genetic profile Ashkenazi Jewish populations carry now. This is the part of the story that actually explains everything else, and it comes down to a demographic phenomenon. Geneticists call a founder effect combined with sustained endogamy, which is simply the practice of marrying within one's own community. When a population goes through a period where it shrinks down to a small number of founding individuals and then rebuilds its numbers primarily through marriage within that same group rather than with outsiders, the genetic signature of those original founders gets locked in and amplified rather than diluted. Every subsequent generation inherits from the same limited genetic pool, which is exactly why specific disease-linked mutations become unusually common in a population, even though those mutations are rare almost everywhere else in the world. For Jewish communities across the diaspora, religious law and social structure created strong and sustained incentives toward exactly this kind of in-group marriage across many centuries. Even while Jewish communities lived embedded inside larger host societies, spoke the local languages, adopted local customs, and in some documented cases did intermarry with surrounding populations. The Erfurt data actually captures this tension directly, showing a community that had already absorbed some external ancestry while still maintaining a genetically distinguishable core. The result across the centuries was not a population frozen in amber, untouched by the world around it. It was something more interesting, a population that absorbed real outside ancestry over time, while a durable ancestral core kept reasserting itself generation after generation, propped up by social and religious structures that made marrying within the community the norm rather than the exception. That mechanism, not mysticism and not myth, is the leading scientific explanation for why a population dispersed across 6 continents for 2,000 years still carries a detectable shared genetic thread today. To understand how deep that thread actually runs, researchers needed to go past the medieval period entirely and sequence genomes from the Bronze Age Near East itself, the world in which ancient Israelite and Canaanite history is set. A major 2020 study led by researchers including Edowen Harney and David Reich, published in the journal Cell, sequenced ancient DNA from dozens of individuals who lived in the southern Levant, the region covering modern Israel, the Palestinian territories, Lebanon, and parts of Jordan and Syria across the Bronze and Iron Ages. What that study found reshaped how geneticists talk about the ancient population of the region altogether. The people living in the Bronze Age Levant were not a single, static, ethnically uniform population. They were themselves a genetic mixture, combining ancestry from earlier local Neolithic farming populations with a significant later contribution from populations related to the Chalcolithic Zagros region of what is now western Iran and the Caucasus, meaning migration and mixture were already shaping the region's population thousands of years before anyone called it the Holy Land. That Bronze Age Levantine genetic profile is not the exclusive ancestral property of any single modern group. When researchers compared it against modern populations from across the region, they found meaningful shared ancestry running through multiple present-day populations, including both Jewish communities and Levantine Arab populations, including Palestinians, Lebanese, Druze, and Bedouin groups. This is not a controversial or fringe finding among the researchers who actually run these studies. It is simply what the ancient Near East was. A genuine crossroads of the ancient world, mixed and remixed by millennia of migration long before any modern political or religious identity existed to claim it. What makes the Jewish genetic story scientifically distinctive is not that Jewish populations are the sole biological heirs of the ancient Levant, because they are not, and no serious geneticist claims otherwise. What makes it distinctive is the specific combination of a documented ancient Near Eastern ancestral core, a 2,000-year global dispersal across wildly different host populations, and a set of cultural and religious practices robust enough to keep that ancestral core statistically detectable across every major branch of the diaspora, from Ashkenazi Europe to Mizrahi Iraq to Yemenite Arabia. That combination, all three factors, held together at once across that much time and that much distance, is genuinely unusual in the historical record of human populations, and it is the actual reason this story keeps showing up in serious research journals rather than only in popular science channels. Every honest researcher working in this field says some version of the same thing once the genetic data is on the table. DNA can measure ancestry, but it cannot adjudicate identity, and it certainly cannot settle a political argument. The same body of research that documents a real ancestral connection between modern Jewish communities and the ancient Levant also documents a real ancestral connection between modern Palestinian and other Levantine Arab communities, and that same ancient population, sometimes at very high proportions in the analyses researchers have published. Geneticists are consistently careful to separate what their data can actually say, which is a statement about shared deep ancestry among multiple modern peoples descending from an ancient, genetically mixed regional population, from what it cannot say, which is anything at all about who holds a stronger present-day claim to any specific piece of land. Anyone who tells you the genetics alone settles that question, in either direction, is going well beyond what the published research actually supports. What the research does support, and what continues to genuinely surprise the scientists who study it, is the sheer durability of the thread itself. Most populations that scatter as widely and for as long as Jewish communities did simply dissolve into whoever surrounds them within a handful of generations. That is the normal expected outcome of genetic drift and intermarriage, and it is exactly what happened to most of the other great ancient civilizations whose descendants can no longer be cleanly traced back to their ancient forebears in any single modern population. The Jewish case did not follow that pattern, and the leading explanation on the table, endogamy sustained by shared religious law, shared text, and shared communal structure operating across completely disconnected communities on different continents, is itself a remarkable finding about how culture can shape biology over historical time. The DNA does not tell you what it means to be Jewish, and no serious scientist claims it does, but it does tell you something concrete and measurable, that the story a scattered people told about where they came from lines up again and again across independent labs and independent methodologies with what is actually written in their genome. So here is where the evidence actually leaves us. A priestly lineage with a testable genetic signature going back roughly 3,000 years. A medieval community in Germany whose teeth revealed a population still visibly diverse just 600 years ago, before something compressed it into the tight genetic profile we see in Ashkenazi communities today. A Bronze Age Levant that was itself a mixing ground of ancient migrations, leaving an ancestral signature that runs through multiple modern peoples of the region, Jewish and Arab alike. And running underneath all of it, one demographic mechanism, endogamy sustained across 2,000 years of exile, that somehow kept a genetic thread traceable across 6 continents when almost no other ancient population in history managed to do the same. The researchers who ran these studies keep saying the same thing when you ask them what it means. The data is real. The pattern is real. The meaning of it is still an open question, and it may be the kind of question genetics alone was never built to answer.